So maybe I can do the presentation because we have well, I'll do it in I say it in Spanish, but tenemos
I can understand a bit better than I can speak. I can
oh,
read and understand a bit, but I'm sorry. I wish I could speak
yes,
better.
no, no, it's okay. Bueno, hoy tenemos la suerte y el honor que nos acompañe el doctor Phillip Lupo, que él es epidemiólogo y que está en la Universidad de Emory y está trabajando sobre, lo que nos va a hablar a nosotros es sobre síndrome de Down y leucemias, ¿no? Así que we want to thank you very much for being with us today, Philip. And well, if you want to, you can have the presentation, no?
Perfect. And can you see it?
Yes, wait.
Yes.
Yes, we can see it. Thank you, Philip.
Perfect. Thank you. Thank you for the introduction, and thank you for the opportunity
Yes.
to come and speak to you today. I obviously was scheduled to talk to you at the end of June, and I had a bicycle accident, which was obviously not fatal, but it was definitely traumatic. And I appreciate the willingness to find another time. I do have the opportunity to work with amazing collaborators in Argentina on our larger Down syndrome Include effort. And our primary interest is in acute lymphoblastic leukemia in children living with Down syndrome. And I hope that gives us insights into the etiology as well as opportunities for early detection. And I'll talk to you a bit about our work throughout the course of the presentation. And just in terms of outline, I'll outline some of the clinical features and challenges in managing leukemia in children living with Down syndrome. That includes disease presentation, outcomes, and toxicities that they experience during therapy. I'll also talk a bit about treatment strategies after a child is diagnosed with leukemia. But I'm really going to focus most of the talk today on the research investigations that we're leading, primarily around germline or inherited genetic variants in relation to etiology and outcomes, and how that information could be leveraged for early detection, which I think is an ultimate goal for us. And I do want to start by saying that the point of the presentation is to engage interested individuals in leadership opportunities for expanding this work in other populations, because as we all know, when we're working in rare diseases, that collaborations are key. And we were having this conversation a moment ago, not just helicopter research, but I think an opportunity to really think and move the science forward as collaborators and co-leaders in these efforts. So what do we know? I don't think I need to spend a lot of time on this, but leukemia in individuals with Down syndrome is a well-described condition. It was actually reported initially in 1930, but systematically studied at first in the 1950s. And consistently, what we see in populations is that children living with Down syndrome have a 20-fold increased risk of developing acute leukemia. The good news is leukemia is extremely and exceedingly rare in human populations. And even though we see this dramatic relative risk, the absolute risk or the cumulative risk is relatively small, in that about 2% of children living with Down syndrome will develop acute leukemia by the age of 5. And if we look at the proportion of individuals diagnosed with either lymphoblastic or myeloid leukemia, about 2% of them also have Down syndrome in acute lymphoblastic leukemia. whereas about 10% of children that are diagnosed with myeloid leukemia also have Down syndrome. There are some unique features of leukemia within children that are living with Down syndrome. And I think it's notable, and obviously a lot of us are interested in the fact that there is both this elevated risk of leukemia, whereas solid tumors are almost absent in individuals with Down syndrome, and this persists throughout adulthood. And then, aside from acute lymphoblastic leukemia, a very common hematopoietic feature is the transient abnormal myelopoiesis, which is this accumulation of clonal megakaryoblast that arise during fetal liver hematopoiesis. And it's driven by a canonical GATA1 mutation. Interestingly, this occurs in almost 30% of individuals born with Down syndrome, but there is, as we know, variable severity, and most of these individuals will regress. But a subset will go on to develop myeloid leukemia associated with Down syndrome. And this typically occurs very early in life, and aside from that canonical GATA1 mutation, there are other acquired mutations beside that driver event. But the good news is these children are very sensitive to cytarabine, which is the chemotherapy that's often used. And because of that, their outcomes for myeloid leukemia are much better than peers that do not have Down syndrome. But most of our work is not in myeloid leukemia, it's in acute lymphoblastic leukemia, or ALL, which I'll refer to throughout the presentation. The age range is similar, at least in terms of onset, to the children that do not have Down syndrome. They're primarily B lineage immunophenotype, but aside from that general observation, we see distinct genetic alterations within the leukemia that is more frequent or less frequent in children with Down syndrome. And that includes CRLF2 rearrangements and JAK alterations. In fact, we led a large study recently funded by the Include effort in the National Institutes of Health, where we were able to do genome sequencing on leukemia tumors obtained from both children with Down syndrome, which you can see here on the left panel, and then tumor sequencing in children with acute lymphoblastic leukemia that did not have Down syndrome. And I think this is actually one of the most striking features in this research, in that about 50%, or actually more than 50% of children with Down syndrome who also have acute lymphoblastic leukemia have these CRLF2 rearrangements, versus only about 6% in a non-Down population. So you can see the disease biology within leukemia is really distinct within this population and different from a non-Down population, which does have implications in terms of therapy, because we know that these cytogenetic alterations are also prognostic and will often drive treatment decisions. So I think this is an important feature that we consider when we think about leukemia risk in children with Down syndrome. And in fact, the cytogenetic features that we see within Down syndrome are typically less favorable than those that we see in a non-Down population, which again is associated with the worst outcomes we see in acute lymphoblastic leukemia in children with Down syndrome compared to those that do not have Down syndrome. So I think many of us on this call are very interested in genetics and the role of inherited genetics on disease susceptibility, both within Down syndrome and in other other phenotypes. And one thing we've done is tried to understand genetic susceptibility to Down syndrome associated acute lymphoblastic leukemia. Now, at the outset of our study, there were a number of publications demonstrating the role of common variants in a range of genes that were associated with acute lymphoblastic leukemia. And you can see those presented in this table. I think prior to the onset or prior to the advent of genome-wide association studies and the identification of these variants, many of us suspected that there would be a gene dosage effect with chromosome 21, whereby an added copy of a gene on chromosome 21 would increase the likelihood of developing ALL. But what you'll see is that most of these variants are not on chromosome 21. In fact, none of them to date have been identified on chromosome 21. The initial genes that we saw in genome-wide association studies of acute lymphoblastic leukemia are ARID5B on chromosome 10, IKZF1 on chromosome 7, and CDKN2A on chromosome 9. Not ones we would expect necessarily on or within an individual living with Down syndrome. So we opted to do a genome-wide association study of acute lymphoblastic leukemia in children living with Down syndrome. And we were able to obtain and conduct a very large study with international partners and amassed a sample size of a little over 500 individuals with Down syndrome who also were diagnosed with acute lymphoblastic leukemia, and almost 1,200 individuals with Down syndrome who did not have acute lymphoblastic leukemia. Obvious case control study in assessing genetic risk. And what you'll see here, I think, is notable, at least in terms of three things. The first of which is that even while only having a little over 500 cases, we were able to see very clear genome-wide associations with acute lymphoblastic leukemia. This is not typical. In other words, I think for those of us who know genome-wide association studies, we typically need thousands of individuals to see effects like this. The second thing that I want to note is that the SNP arrays were deficient in terms of identifying variants on chromosome 21. So you'll see here on this Manhattan plot that chromosome 21 is absent. And that is for two reasons. One of which is the calling algorithms that we use for the SNP arrays could not handle the trisomy 21. But we did do manual calls of those variants that were detected on 21 and still didn't find anything that was consistently associated with leukemia. So that was of interest. But what we did find, again, were several associations that were both strong and consistent with what we would see in a non-Down population. So in other words, we see the same associations in a non-Down population as we do in a Down population. But I think what was really compelling to us is that the effect sizes were much stronger than we typically see. I think, again, for those of us who work in genome-wide association studies, the effect sizes are not typically this large. So that was of note to us. So we really wondered if there was a biological effect where inheriting these variants in the background of trisomy 21 is what would increase susceptibility. So the next thing we did is look within subtypes to see if these associations were consistent. And notably, the strongest association that we saw with CDKN2A, where we saw this this missense variant is that it was associated with all subtypes of ALL within Down syndrome and not significantly different. So again, what we did see is this genetic effect was consistent across subtypes. But this work, while it was informative, I think really just, as most research does, led to more questions. And this was around the time that the NIH was rolling out Include, and we were able to work with Joaquin Espinosa and others to assemble additional samples with the idea of doing whole genome sequencing. And that would give us information on more rare variants, as well as structural variants, and hopefully variants on chromosome 21. These analyses are currently underway. And interestingly, to date, in our whole genome data, we did see several of the same patterns that we saw with the SNP array data. But I want to draw your attention to a new locus that we've identified with the genome data that could be a unique susceptibility variant within Down syndrome, and that's a locus in TPX2. And you can see here the differences in allele frequencies between cases and controls, but also the effect size is quite dramatic. So again, I think the whole genome sequencing data is giving us new insights into novel variants associated with leukemia that suggest that leukemia risk, in part, not surprisingly, is driven by inherited genetic variants within Down syndrome. There is no evidence that there are other signals within the region. This is a regional association plot of those variants in TPX2. And you can see here that our leading variant is in linkage disequilibrium with other variants in the approximate area. And again, consistent with, I think, a strong association. And while I think these findings are important, I do want to highlight that this is only the beginning of this work for a couple of reasons. One thing that we discovered, as I've noted, is that really non-Down associated acute lymphoblastic leukemia susceptibility looks very similar to that we see in a non-Down population. So what we suspect is that, again, that trisomy modifies the penetrance of these variants. They're the same variants that we see in a non-Down population, but the effect sizes are much stronger. So we do think that there's an interaction between being born with both trisomy 21 and these variants. But I do think there's more to discover in terms of the genetic architecture of leukemia in children with Down syndrome, which we hope the whole genome sequencing data will yield. And I think the finding within TPX2 is a notable one. And we do think that there is much more to discover on chromosome 21 with deeper sequencing, as well as some of the long read sequencing that we're currently conducting in this population. But I think most of us are really interested in not just identifying genes that drive leukemia risk, but if we could leverage this information to potentially predict children at birth who are to go on to develop acute lymphoblastic leukemia with the idea of doing improved surveillance. And the reason this is possible is we think the biology behind leukemia. And that's really a delayed infection hypothesis that leads to leukemiogenesis. And this was proposed by Mel Graves, for those of you who might be familiar with leukemia research. But the idea is that children are born with pre-leukemic clones. And we do see this in blood spots, and we see this in cord blood of children who go on to develop leukemia. And there is some sort of event, some sort of pathologic immune response to infection or some other stimuli that suppresses hematopoiesis and allows the expansion of these clones. And there's a selective advantage of these clones, which will go on to develop into overt leukemia. And again, this is consistent with animal models, as well as, again, the identification of these leukemic clones within blood spots obtained at the time of delivery. So our idea is really taking this biological phenomenon and trying to backtrack these pre-leukemic clones, or identify these pre-leukemic clones at the time of birth, with the idea that this could give us insights into the natural history of acute lymphoblastic leukemia. And it could also allow us to understand the subsequent events that lead to overt leukemia. And those are things like infection, again, related to immune response. And then hopefully the idea would be to leverage this information to promote early screening for those individuals that go on to develop acute lymphoblastic leukemia that might help in earlier detection or better disease management. And the good news is, aside now from just identifying clones at birth, is we can actually identify those fusion sequences that I pointed to in that somatic or tumor sequencing study. And that's especially important for the CRLF2 rearrangements. And you might remember that about 60% of children with Down syndrome that develop leukemia have these CRLF2 rearrangements in their tumors, which we think can be identified within the blood spots. But there really hasn't been a study to date that has extensively studied the presence of these rearrangements in individuals that are born with Down syndrome. So what we're planning to do, and what we propose to do, is develop an international collaboration to think about identifying the prevalence of the CRLF2 rearrangements at the time of birth. And we would hope to use archived blood spots or some sort of sample collected at the time of birth, and then couple that with the germline genetic variants that we identified in our larger studies to see if the presence of these rearrangements in blood spots coupled with polygenic risk scores could be used to predict the children that are most likely to go on to develop acute lymphoblastic leukemia. And it's, again, a very simple study design. And we would think about individuals or neonates that are born with Down syndrome, some of whom go on to develop acute lymphoblastic leukemia and some of whom don't. And obviously we would have to have that kind of follow-up data to do so. And we do in several states that we've started to collaborate with. And those rearrangements can be detected through droplet polymerase chain reaction, or PCR. And we've demonstrated that that can be done using blood spots. In other words, we can use PCR to identify these CRLF2 clones. And then using the blood spots, we would also perform the germline sequencing and then compare the frequency of both the clones and the polygenic risk score from our previous studies in cases versus controls. So again, most of this work is very preliminary. And when I was talking to Graciela at our last Down syndrome Include meeting, we thought that it would be an opportunity to think of expanding to populations that have traditionally been excluded from these larger efforts. And again, I think this would be the opportunity and the timing at which we could really utilize the input and leadership from individuals that might approach this from some different perspectives. So just as a reminder, I think it's very notable, and I don't know that we can say this, or maybe we can say this for all co-occurring conditions in children with Down syndrome, but acute lymphoblastic leukemia that's associated with Down syndrome really has a unique presentation that is reflective of underlying differences in the biology of leukemogenesis. And I think that's important because we can use that information to think about prediction of risk in this population. And while the established variants that we see that are associated with leukemia are the same that we see in the general population, it's notable that they actually exert a stronger effect in terms of predicting risk. So I think that's, again, notable in the power of a polygenic risk score. And furthermore, we're finding novel variants that are also associated with leukemia risk, which I think will strengthen our ability to understand leukemogenesis and think about prediction as well. And the fact that the leukemia subtypes that we see in acute lymphoblastic leukemia in children with Down syndrome is characterized by these fusions and other distinctive transcriptomic profiles, I think can be leveraged at the time of birth if they can be identified in small proportions to, again, predict the risk of leukemia. But we're at this crossroads now where we really need to expand this and test these hypotheses. So this work, I think, again, has implications in terms of understanding pathology and pathogenesis, but it can also inform not only treatment responses and choices, but I think it gives us an opportunity to better risk stratify children diagnosed with leukemia that also have Down syndrome. And then hopefully it can be used as a tool for surveillance and early detection. And to date, we've had a number of colleagues help us in this effort that have played key leadership positions, and the funding has really benefited from a number of foundations as well as from the NIH. And with that, I wanted to leave some time for questions, and I'll say two things. I'll leave my email here if you would like to follow up and have a conversation at another time. But I'm also planning to share these slides with the group with the idea that this information might be useful if you're interested in taking some of these ideas and moving it forward on your own, or if you're interested in thinking about opportunities to collaborate more extensively. And with that, I'll end and I'll pass it back to you, Viviana.
Yes, it's okay. Yes, that would be fine. Because I think that most of the people that can help or could be interested in helping, they are not here now, but we can share the presentation.
I would be happy to do that. There's no proprietary information or anything that needs to be held confidentially here.
And I don't know if, no sé si alguien tiene alguna pregunta o quiere hacer alguna pregunta.
I would like to ask the question, how many blood spots do you need for this test?
It really doesn't need to be a lot. I think if we could identify 50 blood spots in children that go on to develop leukemia and 50 blood spots in children that do not, that would be a very strong proof of concept for this. Because again, I think what we're hoping to demonstrate is a very powerful signal. So we don't need thousands to do such. I think, again, dozens would be ideal.
Okay, because all of our children have the Good test down for hypothyroidism and all that disease, and they kept some blood spots. And can we use that to send that to you, for example?
That would be amazing and ideal, and we would be happy to explore that possibility, because it's, again, I don't think it's not a scenario where we need a lot of material to identify these leukemic clones in blood spots. And we do think that they're going to be relatively prevalent, especially in those that go on to develop Down syndrome.
And did you have any study done um, in in uh, in the sample in the sample that you use for transplantation when you use cord blood?
I'm not presenting any of the information here, but we actually have a study through the Children's Oncology Group where we have identified individuals who have banked cord blood, and we have been able to obtain that cord blood, and we have used that to identify these translocations. So that is another opportunity. If there is banked cord blood, these events can be measured very well in those samples.
Okay.
I do you have a but because the problem would be the ethical
Ethical aspects.
aspects, no? Because in order to be able to bring it here, it's there if that is the implementation for example.
No, perhaps you need an principal investigator here, and he can be the one that directed the project here. And
that would be the idea. You know,
yes,
the model
it can
that
be approved,
that's right.
but any
Okay.
ethical committee, and he will
Yes.
responsible to ask for the samples, to speak with the public, for the parents, the informed consent, and to send the samples to United States. But that costs money, so perhaps it would need some grants to
Funding.
do that.
Yeah.
Especially the sending of the sample.
Yes, and that would be the model. I think there's one of two models that we could explore. One of which would be that exactly. We would identify a principal investigator who would like to take a leadership position in the project and collaborate in a way where we could also send funding for the collection of samples, for processing and shipping. The other model is these are technologies that I know that one could do in Argentina easily. So the other option would be sending somebody to train individuals if needed to do this work there. So I don't think it would be, if there's a problem with shipping samples, they could also be processed in the actual assessments could be done in Argentina.
Okay.
Yes, and perhaps we have to standardize the technique. So we use the same kits and everything.
Right. I think that would also be a very good possibility. And yeah, and I do recognize that good collaborations require three things, I think. One of which is a real engagement from the principal investigator that has an interest. The second is, I think, ensuring that principal investigator has a prominent role in the publication of these results. And the third thing that we acknowledged is that we would need funding to make sure this could be possible. And I do think that the Include program is a very good example of, while we're trying to work out some of the details with our new administration, how this is going to work, but there's still opportunities for funding research in an international way, especially related to Down syndrome. There are also foundations that I think are very interested in this work.
Yes, we have to think about that.
Yes.
Perhaps at university we can perform the test to do it locally.
I think that would be a great option.
Protocol, yes, with the same protocol, so we don't have to send the samples abroad.
I think that, I mean, there's no magic involved in these things, and I don't.
No, it's a digital PCR, so I think we can do that.
Exactly. So again, I think if there are specific components related to the detection of these specific translocations or clones, we could send the protocol, and you know, a lot of this is just modifying things that come off the shelf. And we could also, you know, use this as an opportunity to come for a visit and make sure that processes are harmonized, or if there was a technician that wanted to come to the lab here, I think there would be many opportunities to think of.
Sometimes what we do is sometimes you send us a sample, so a positive and a negative sample, but that's
a really good point. Yes.
Blind, and we standardize the sample here.
Mm-hmm.
No, Vanina is Vanina Medina from Biomed is writing to me. she's watching this presentation by YouTube, so she's really enthusiastic.
Oh, good. I think that there in Graciela, this was our point. I mean, I think that I hope my intention is clear that this is not a scenario where I want to come and say, please just send me your samples.
No.
The intention is really to think of a true collaboration where samples, if sent here, that's fine. I don't think it needs to happen here, especially if that limits opportunities. And again, I don't think there's any need for the processing to be done in a single lab in a single place. So.
And I think it would be a great opportunity also for a PhD thesis. So.
Yes.
Actually, you know, that could be really interesting, because I do think one thing that, and I think this is true for other co-occurring conditions in children living with Down syndrome, is even doing some of the groundwork and thinking about opportunities for looking at the somatic landscape of leukemia in your local population. Because again, you know, the other thing that we have seen is that several of these disease features do vary by genetic ancestry. So it might be interesting to see if there's differences in proportions of some of these things. And again, if there was an interested PhD student, this wouldn't require sending samples here again. This could all be done locally, and we could, but I think it could be a collaboration where if we did have funding to support the work, that it could be right. It could be conducted locally.
Yes, and we can also find support here. We can work on that. If we have a strong project, I think we can work on that.
I think that's an amazing opportunity. I have found that there is nothing like an eager PhD student to move a project forward in that regard.
Well, we have to think about it.
Let's do. Well,
Yes.
and again, that was the intention. You know, I didn't want to just come to solely present results, even though I enjoyed that. The intention is to think about an opportunity for expanding this work, because I do think that, again, we can acknowledge that leukemia isn't extremely frequent in children living with Down syndrome, but it is an important diagnosis that these individuals face, and especially for ALL, where we see such worse outcomes. I think it's important to think of strategies for improving our knowledge of this particular disease.
Yes, I think, especially the parents, I think they will be interested if we can look for a more personalized treatment.
Yes, absolutely. And that, I mean, I do think oncology, and you know, maybe that's because this is the space I work in, but I do think there is a growing awareness of targeting certain somatic features for therapy. So, you know, precision medicine in this case, I think, has some real opportunities, because again, you're targeting certain somatic features.
Yes. Yeah. Well, we have homework now.
Yes. Well, and I would love to come for a visit. Yes.
Yes, it's very. Here we have we at CEMIC, we are the head of the ECLAM, that is multicentric study in newborns that has around 60 years in all Latin America. So there are many people who can maybe be interested in collaborating, but that would be the difficulties are those two with ethic committees and all that. But
that's not a problem for us. I have been president of our ethic committee for 3 years.
I know that.
So
I know
I
that.
have clear.
No, but we
can do something collaborative here in Argentina, and not only one hospital or only one university. We can do it together, and
yes,
I think it would be interesting.
In 2003, we had a grant from the NIH with the ECLAM that it was about clefts, oral clefts.
Yes, I'm actually very familiar with this because I do also work in structural birth defects, and I'm familiar with the ECLAM work in clefting as well. So that is a very notable research.
Yes.
Okay.
Yes, but we can look for some local initiation like Funded or something about CONICET, where we can find some money to do that. So we have to think about it.
Yes.
Okay, I'm happy to keep up the conversation.
Sí, pero se puede, desde la UCA se puede.
Desde la UCA vos decís que sí, te van a dar.
Yes,
yo no sé en CEMIC, viste cómo viene.
No, because we have our institute, our research institute is Biomed. It's UCA CONICET. So
no, CEMIC también. CEMIC is CEMIC CONICET, but the problem is the grant, if they give you the grant. That's the problem.
That's why we have to present a very strong project to do that.
Yes.
And I think it's 3 or 5 determinations only for the molecular fingerprint, isn't it?
Okay.
That's right.
And surely we will. It would be very difficult for us to do genomic sequencing. But yes, just the specific determinations, I think we can do that.
Mm-hmm.
Well, I'm excited about this.
We have to think about it.
Yes, please do, and let me know if you have questions in the meantime. And Graciela, I will send the slides back to you. I think you have them already, but
yes, I have them in English and Spanish, and I sent it to Viviana in English and Spanish.
Perfect. Well, I'll thank you so much. It was a pleasure.
Thank you very much for your time. We get in touch. Okay.
I'm looking forward to it. Take care.
Nice to see you again.
Bye. Good to see you. Take care.
Thank you. Nice to meet you.
See you next year here in Argentina.
Yes, absolutely. I'm excited.
Okay.
Bye-bye.
Bye-bye.
Bye.
Bye. Bye-bye.